So today I would like to talk about the structural and functional characterization of the SARS-CoV-2 receptor binding domain. So as we heard already from Charlotte, there are non-structural proteins, but now I will talk about the structural proteins. And there you have in the coronavirus, for example, the envelope protein, the membrane protein, but also the spike protein. And this spike protein is a very important protein because via this interaction with the ACE2 receptor, the spike protein is infecting our cells. And if we have now a closer look at this spike protein, you can see in this animation here or in this model here that there is a trimeric protein and consists of 3 times this S protein. And this S protein has a domain, the so-called receptor binding domain, where I will talk today about. And this domain, you can see we have also then 3 times in this trimeric protein. 2 are in a down conformation, but 1 of them is in an up conformation. And you can see from the side in the model that this is really looking outside of the protein. And this is responsible for the interaction between the ACE2 receptor and the coronavirus. So this is a very important domain of this S protein. If we look then into more detail in this domain, you can see that we have that the domain is 222 amino acids large and they're roughly about 30 kilodaltons heavy. It is glycosylated, so it has 2 N-glycosylation sites and most probably 1 O-glycosylation sites. And it was also shown already that this glycosylation is part of an epitope for neutralizing antibodies. And in general, you saw that this RBD is really looking outside of the spike protein. So this is really normally the target for the neutralizing antibodies and thereby a very important domain. Next to this, what is also known already from literature, that if you cleave the glycans off and you just analyzed or have just coronavirus with a non-glycosylated spike protein, then the infectivity goes drastically down. So because of this importance of these domains, there are more and more companies out which are producing recombinant RBDs for diagnostic or vaccine development, but also for research purposes. However, most of these recombinant produced RBDs, they are not well characterized. So normally they come just with the SDS gel, but more information is normally not known about them. So therefore, we collaborated with a company which is called In Vivo, and this company is producing the RBD either in CHO cells, but also in HEC293 cells. And together with them, we wanted to do a complete structural and functional characterization of this RBD domain. So this is our analytical strategy, what we applied for this. So on one side, we analyzed the intact protein, but also we did a dissection of the glycans to get information of the combination of the N-glycans on the intact level and the O-glycosylation, the putative one. This was analyzed by c-MS, but also by multi-insorstic-MS. And next to this, we also analyzed the glycopeptides and the released glycans to get a confirmation of our findings, but also a quantitative information of the glycoforms. After the structural characterization, we also wanted to characterize the RBD domains on the functional level. And therefore, we wanted to see if the antibodies which are built normally against the spike protein are also binding to the RBD. But also on the other hand, obviously, we wanted to see if this RBD domain is binding to the ACE2 receptor, which is the main function of the RBD. And this way, we get in a complete structural and functional characterization of the RBD domain. If we look now how we did the structural characterization first, and we just analyze the intact protein, you get a not so complex spectrum for the CHO cell. However, if you look at the HEC cell produced RBD, you get a very complex spectrum. And if we tried in both spectrum to assign any glycoforms, this was really hard. And also we saw that there might be additional proteoforms on the protein backbone. So for this reason, to start with, we removed completely the N and the O glycosylation, which gave us then, or should give us then, a theoretical mass of 25,914. However, if we look then at the CHO cell produced RBD, we see that we get an elevated mass, which has a delta of 119 Dalton. And after this RBD domain has a free cysteine, our first thought was maybe it's a cysteinylation. And indeed, if we analyzed then the protein under reducing conditions by c-MS, but also by multi-MS, we could see that this modification disappears, and therefore we assume that this is a cysteinylation. If we look then at the HEC cell produced material, you can see that the spectra looks already much more complex, and you can also see a +42 Dalton variant. but also in overall the mass is 230 Daltons heavier. So if we subtract the cis-denylation, what we also had on the CHO cell produced material, then you can see that we still have a shift of 110.9 Dalton. And if you look it up in literature, this fits quite nicely to a pyroglutamate. And therefore, we did them some multi-insource decay fragmentation. And you can see that we get a really nice sequence coverage of the N-terminus. And indeed, we could confirm additional pyroglutamate at the N-terminus. And also, we can confirm this by other bottom-up approaches. Next to this, as I told, there is also this plus 42 Dalton variant, which we can confirm with bottom-up approaches to acetylation on the protein backbone. And also, we could observe a 730 Dalton bigger signal. And this was looking like glycans. So then we looked again in this enzyme, what we used for the O-glycosylation, to remove the O-glycans, or the O-glycor. And if you can see, this enzyme is only able to remove core 1 type glycans and core 3 type glycans. So this we could clearly then assign to, or we were thinking that this might be core 2 type glycans. And indeed, if you look then at the released O-glycans for the CHO cell produced material, you see only core 1 type glycans and a very minor amount of core 2 type glycans. Whereas in the HEC produced material, you can see also core 1, but also a very big amount of core 2 type glycans. So indeed, we could assign this then to a RBD domain with core 2 type glycans on, which cannot be removed by the O-glycor. So next to this, after we know already that there is O-glycosylation at the RBD domain, we wanted also to localize it. And certain approaches or certain articles which are already out about the intact spike protein, they were never be able to localize this O-glycosylation because of their close proximity. So this was not possible with bottom-up approaches, but also with MSMS, they were not successful. And for this reason, we tried another approach. So we used the so-called O-protease, was called operator. And this O-protease is cleaving either N-terminal to the O-glycan, and so it would cleave either N-terminal to the threonine or N-terminal to the serine. And in the CHO cell produced material, you clearly can see a cleavage in N-terminal to the threonine. So there is localized at the threonine, the O-glycosylation. And in the HEC cell produced material, we clearly can also see it on the threonine, but also we saw an additional signal again. And if we look again in the specifications of these enzymes, like before, this enzyme is not able to cleave the core 1 and core, this can only cleave core 1 and core 3 type glycans, but not the core 2 type glycans. So we have an uncleaved RBD with core 2 type glycans on. Next to this, we also wanted to characterize then the N-glycans, and this, the best thing is to do this then on the intact level also, that we also get the information on the combination of the N-glycans together with the O-glycans. And for this reason, we applied a dissection of the glycans. So first we analyzed it on the intact level, then we removed the sialic acids, we removed the galactoses, and in the last step, we removed the N-glycans and just looked at the O-glycans on the intact level. Next to this, for confirmation and quantification, we also analyzed the RBD on the glycopeptide level and also on the release glycan level. So here are again the spectra what I showed you at the beginning. So the HEC cell produced material in the upper one and the CHO cell produced material in the lower panel. And if you treat this now with a galactosidase in the sialitase, then you can see that we really can simplify the spectra and get only the core N-glycan structures, which are then very easy to assign with the information on the protein backbone. And if we do this now for the CHO cell produced material, you can see that we easily can assign all these core N-glycan structures. But if we remove the sialic acids and the galactoses, in the next step, we only remove the sialic acids and we can assign all the galactoses. And in the third step, we analyze it then on the intact level, and you can see now the spectra is really easy to assign. And what you also can see is that we have really in the CHO cell produced material very minor amounts of sialic acids on the N-glycans, but therefore we have very high antennary and also several Lagnac repeats. In contrast, if we look now at the HEC cell produced material, we can do the same. So we can assign after galactose and sialic acid removal the core glycan structures. And the interesting thing of the HEC produced sample is that we can also find some antenna fucosylation. And next to this, if we remove then only the sialic acids, you can see that we can assign now all the galactose structures. And in the last step, we can now assign this really complex spectrum on the intact level. And as I said already, here we have some antenna fucosylation glycans, which we could not see in the CHO cell produced sample. And another difference is that we have very heavily sialylated glycans. And also we can see that we have normally not this large antenna or this high antennarity, and also no Lagnac repeats in the HEC produced sample. After I told you already, also we analyzed this protein now on a glycopeptide level and release glycan level. We used an approach which was developed by the group of Albert Heck, and this is they developed an algorithm where you can simulate a spectrum from this glycopeptide and release glycan levels. And in case of the CHO material, which has a very minor amount of sialic acids, you can see that the real spectrum is fitting very nicely to the in silico simulated spectrum. However, if we look at the HEC produced material, you can see that the simulated spectrum is shifted to the lower mass range and the correlation is not so good. So this might have two reasons. On one hand, maybe the algorithm is assuming that the glycans are equally distributed. However, maybe the sialylated glycans are not equally distributed over the protein. And on the other hand, you can also lose during bottom-up approaches some sialic acids, but also obviously there is an ionization bias coming from the negative charge on the sialic acids. So for this reason, it is also important to analyze glycoproteins not only by release glycans or glycopeptides, but rather on the intact level to avoid skewing of the data in any direction. After I showed you now how we functionally characterize the RBD domain, I want structurally characterize the RBD domain. I want to show you how we functionally characterized it. And for this, we wanted to see how the RBD domain is binding to antibodies especially from patients which had already a coronavirus infection. And therefore, we used a serum of 12 antibodies and extracted captured the anti-spike antibodies, coated an ELISA plate with it, and determined the binding of the RBD domains to these antibodies. And as you can see here, for the CHO produced material, there is a clear binding of the CHO produced material to the antibodies, which in comparison to the negative control. However, if you remove the complete glycosylation, you can see that you have a slightly elevated level, but the same effect you can see also in the negative control, and this might come from all the glycosidases what we used. So in this case, most probably the glycosylation does not have an influence on the binding. A similar effect we could also see for the HEC cell produced material. So also we have the same binding like in the CHO cell produced material to the antibodies, and we could see also the slightly elevated level of the completely deglycosylated protein, which was also visible in the negative controls. And after I told you the main function of the RBD is to bind to the ACE2 receptor, obviously we also wanted to test the binding to this ACE2 receptor in vitro. And therefore, in this case, we coated the ELISA plate with the ACE2 receptor and determined the binding of both, so the CHO and the HEC cell produced material. And what you can see nicely in this graph is that both of the domains, so the green and the blue one, are the complete intact RBD produced in HEC and CHO, and they show a very nice binding and a very strong binding to the ACE2 receptor. But you can see a slight difference, so the HEC produced material binds slightly less strong to the ACE2 receptor, and this might be due to the different glycosylation. So either the antenna fucosylation or the sialylation might decrease the binding. Next to this, if you completely deglycosylate this RBD domain, which is shown here in brown and purple, you can see that the binding is even lower. So somehow the glycans are involved in the binding to the ACE2 receptor. If we look now, or if we compare now the binding to the intact spike protein, but also just to the S1 domain, which contains this receptor binding domain, you can see that both RBD domains have a much stronger binding than the intact spike protein, and even a stronger binding than the S1 subunit. And this might be due to the different accessibility of the RBD and can also be beneficial in certain applications. So to summarize, so we analyzed RBD produced in CHO cell, but also in HEC cells. And both of the RBDs showed a strong binding to ACE2 and also a binding to the anti-spike antibodies. We could confirm a cysteinylation on the free cysteine in both of the CHO and HEC cell produced material. Also, we could localize the O-glycosylation at the threonine 323. And if we look now specifically at the CHO cell produced material, you can see that we have a low amount of sialic acids, nearly exclusively core 1 type O-glycans, and a high number of antinerity and Lagnat repeats. Whereas in the HEX cell produced material, we have a high amount of sialic acids. We had a mixture between core 1 and core 2 type O-glycans. And we could observe antenna fucosylation, which we could not observe in the CHO cell produced material. Next to this, we could observe on the protein backbone also some acetylation and also some pyroglutamate and the N-terminus. So before I finish now with my talk, I also would like to show what else are we doing in our group. So we are not only characterizing proteins, but also we are characterizing the antibodies and the glycosylation of the antibodies against the spike protein. And in this case, we have a technician, Jan Nauta, which is doing this in the lab. And he is capturing from one serum, from one serum of one patient, the total IgGs with protein G beads, and with the spike protein, he's capturing the anti-spike IgGs. Afterwards, he is digesting them and analyzing them via LC-MS. And we have another student in our group, Tamas Pongratz. He is then doing the data evaluation, and he wants to see if there's a difference between the total IgGs and the antigen, so in this case, the spike-specific IgGs. And this is now an example of his data. So in the upper panels, you can always see the total IgGs. In the lower, you can see the antigen-specific IgGs, so the anti-spike IgGs. And if you look now, for example, here to the G0F form and the G1F form, you can see that in the total IgG, they are clearly visible, whereas in the specific IgGs, the G0F form disappears completely, and you can see a strong decrease in the G1F form. The same effect you can see if you look at the sialylated structure. For example, if you look at this glycan structure, which is visible in the total IgGs, this cannot be observed at all in the antigen-specific IgGs. If you look then at patients which were administered to ICU, then you can see if you look at their total IgGs, the fucosylation level stays nearly the same over the time. Whereas if you look at the specific IgGs, so the anti-spike IgGs, you can see that at the beginning when they are administered to the ICU, the fucosylation levels are very low. And then over the time course, when the patients get better, the fucosylation level normalizes. And this resulted also in a publication where we could link the fucosylation of the IgGs with the severity of COVID. And if you're more interested, you can have a look. And with this, I want to thank my supervisors, Elena and Manfred. I want to thank the collaboration partners from MVivo, Tim and Sophia. And on the other hand, also the collaboration partners from Brooke, Detlef, Anja, and Stuart. And thank you for your attention.